Hello, everyone. My name is Kishore. I am a postdoctoral researcher working with Professor Herbert Levine at CTBB Northeastern University. Uh, you can see the fall scenery in my background. Northeastern is a very beautiful campus. Boston is a very beautiful city. Please do visit if you ever have a chance. So I would like to start by thanking Tommaso for inviting me to present at this colloquium. I'm very happy to have an opportunity to talk about my work and also have a platform to thank ESMTB for the awards that they have kindly given me. So, given the context, I thought it will be good to talk about my PhD thesis work. During my PhD, I was interested in understanding design principles of gene-regulated networks, particularly in the context of cancer metastasis. And that's what I'm going to be talking about in the next 40 minutes. So metastasis is a very deadly, dangerous disease. Briefly, what happens in metastasis is that cancer cells start to invade different tissues and different organs in the body, thereby causing multiple complications leading to multiple organ failure. And obviously, they are associated with majority of mortality related to cancer. But what is not so obvious immediately and so interesting about metastasis is that it's a very inefficient process. Once the primary tumor matures, whatever that could mean, there is a lot of dissemination happening, which is to say that there are a lot of cells that are constantly detaching themselves from the primary tumor cell and trying to get into the bloodstream, trying to get into a secondary site, which is more suitable for their growth, right? But a very small percentage of cells are actually able to do so. And the main reason for that is the path that metastatic cells have to take, right? So at the primary tumor, there are already multiple biochemical and biomechanical signatures. Environment keeps changing constantly. You have different layers of environment, but especially when cells start to migrate in the primary tumor, in the bloodstream, and in the secondary tumor, their environment keeps changing constantly, right? So in order to survive, metastatic cells have to maintain a balance between two properties. One is the ability to switch to a different phenotype. A phenotype of a cell is the collection of functions that allows it to survive or perform a particular behavior in a given environment, right? So since the environments are changing constantly or at least dynamically, the cancer cells have to have an ability to change their phenotype accordingly. That's called phenotypic plasticity. And the phenotypic robustness is the other aspect of it, where uh, cancer cells have to maintain the set of functions or the set of phenotypes that are viable, that are good for the current environment, as long as the current environment passes, right? Once the environment changes, they might change the phenotype, but as long as a particular set of environment is maintained, they have to maintain a particular property, right? And a balance between these two properties is one of the fundamental driving forces behind successful metastasis. So the question for the cellular phenotype is to be or not to be, right? So imagine you have a group of cells, heterogeneous cells with different phenotypes, and these cells are now exposed to a perturbation. The perturbation could be biochemical, environmental fluctuations, or mutations, which can happen a lot in cancer, right? Now, if the signal is relevant, then if the signal is not relevant, then they have to maintain their phenotype. The population heterogeneity has to be maintained, broadly speaking. That's called robustness. And if the signal is relevant, right, then they should make a decision to change their phenotype, thereby changing the population heterogeneity as well, right? So for the longest time, it bugged me how these two processes, which seem so different on paper, right? One is about changing their phenotype, the other is about not changing their phenotype. How can these coexist together in the same cells? And what driving forces are there that allow these coexistence, right? So we're going to look at this problem through the lens of epithelial mesenchymal plasticity. This is just one cellular process, which is very important, very crucial underlying cancer metastasis. And briefly speaking, the cells at the primary tumor or the secondary tumor sites, they have a phenotype called the epithelial phenotype, which allows them to adhere to their surroundings, adhere to the surrounding cells, and grow, right? Whereas while in migration, they have a more what is known as a mesenchymal phenotype, which allows them to detach from their surroundings and migrate and sometimes invade through the barriers between the tissue and blood vessels, right? So EMT is a good system to study phenotypic robustness and plasticity. And the reason is actually pretty straightforward because cells undergoing EMP, they are maintaining their epithelial phenotype at the primary tumor site and the secondary tumor site, right? And as they are going through the migration, they are maintaining more mesenchymal or more hybrid phenotype. So they do have robustness properties. And similarly, they also have the ability to change their phenotype given the requirement, because before entering secondary tumor site, they have mesenchymal phenotype, but as they enter to a secondary tumor site, they have to regain their epithelial phenotype. Similarly, when entering the bloodstream to migrate, they have to change from an epithelial to a mesenchymal or a hybrid phenotype. So there is a switching happening between these three broad classes of phenotypes. The epithelial, again, as I've said, is adhesion and growth. Mesenchymal is more invasion and migration. And this hybrid phenotype, which has the properties of both epithelial and mesenchymal, has been proposed to and shown, in fact, to be the most metastatically fit phenotype among the three of them, which has the properties of both epithelial and mesenchymal phenotype, right? So the question that we ask is, how do cells undergoing EMP achieve plasticity and robustness? And when we talk about phenotypes, right, phenotypes are driven by the underlying biochemical molecules. The expression levels of different biochemical molecules, different sets of biochemical molecules, is what determines which phenotype a cell can exhibit. Right? For example, the epithelial phenotypes have a bunch of molecules which allow them to generate these integrins or adherents, catherins, etc. They allow them to attach to different surfaces. Whereas mesenchymal cells have markers or protein expression that allows them to migrate and invade by degrading extracellular matrix and so on and so forth. So any change in the phenotype or any not change in the phenotype has to involve the expression levels of these molecules. And that is regulated by what are known as gene regulatory networks, right? So gene regulatory networks are essentially the expression of proteins is governed by expression of genes, right? And at a transcription level, these expressions are interconnected in that the expression of one gene, for example, snail here, can suppress or activate expressions of the other genes, right? So when we take these sort of regulatory interactions between gene expressions together, we get these gene regulatory networks. And the advantage of EMP system is that it has been studied extensively for over 2 decades now. And that led to the discovery and construction of a lot of these gene regulatory networks. Each network was constructed in a different context, but one common feature across all of these networks is that they support the switch from an epithelial to a mesenchymal phenotype, and therefore they support EMP, right? So what we did was we made a collection of multiple such gene regulator networks and of different sizes, of different complexities, and we asked the following questions. The first question is, how robust or plastic are the phenotypic spaces that are emergent from these gene regulatory networks? Secondly, are there any common features across these EMP networks, right, which allow the phenotypic space to be plastic or robust? Third, can these features act as governing principles, in that if you take those features out of these network structures and put them in a different context, different network, then would those contexts or those systems still show plasticity and robustness characteristics? Now, we could be looking for these principles at different levels of information, because when we are trying to simulate gene regulator networks or model gene regulator networks, we can incorporate information at different layers, right? We'll incorporate the information at the topology level, how each node is interacting with the other, regulating the other. We can also incorporate the information at a kinetic parameter level, and which is the interaction strengths, production rate, degradation rate, and so on. And we can also incorporate noise levels, right? But what we're trying to do is try to understand features that might be common across different contexts, right? And in cancer, it becomes even more important because there is accumulation of a lot of mutations. And these mutations can affect these different layers of information differently. But arguably, the effect of these different contextual information or mutations is the least on the topology, right? So if there is any behavior that has to be consistently maintained, it is more likely that it's ingrained in the topology than any of these in other layers of information. So we want to ask, does the network topology have a significant control over robustness and plasticity? And to answer that question, we need to simulate the system, the network, and figure out what are the phenotypic spaces or landscapes allowed by the network topology. We can do this in two ways. One is using the standard Boolean approach, where we're not really worried about what are the interaction parameters, what are the production rate, what are the degradation rate. We are mostly just interested in what is the nature of the regulation, right? So here is a simple example of that, where each node here is a gene, and then we only care about whether the gene is on or off, -1 or +1. And then we only care about the nature of these interactions. So we either care about its activation or inhibition, right? So again, the signs of each interaction are also +1 or -1. And the way we simulate these systems is that we start from a random collection of initial conditions for all the nodes in the network, and then let the system evolve to a steady state and use the steady state as a representative of the phenotype that we observe experimentally. The second way to simulate this is to take an ODE-based approach. So for any gene regulator network like this, we can construct a system of ordinary differential equations, each ordinary differential equation telling us how the expression of each node changes over time. And each ODE will have different parameters. You have the production rate, you have the degradation rate, and you have these interaction terms which have the corresponding interaction parameters. Now, instead of simulating these at specific sets of parameters like we would for any specific systems, since we want to look at the effect of the network topology, we sample parameters from a large parameter space, right? And we simulate the network at each of these parameter set and let it evolve to steady state, use the steady state as the representative of the phenotype, and ask what are the possible behaviors allowed by the network across this large parameter space, right? So these are the two simulation formalisms that we use interchangeably, and both of them are doing essentially this, that they are generating a heterogeneous population of cells. In the Boolean case, each cell starts from a different initial condition. In the ODE case, each cell starts from a different parameter set, right? But all the cells have the same GRN. Now, using this simulation formalism, using this set of simulation formalisms, we first wanted to quantify how much, how phenotypically robust and plastic are the emergent phenotypic spaces. So first comes the definitions of what we consider plasticity and robustness in this particular context. So plasticity, as I've mentioned before, is the ability of cells to switch between different phenotypes. And this is very clearly defined in the ODE context, where each cell here has different parameter sets, and you could either, so each parameter set could either be a monostable or multistable, right? And assuming that we are not changing the parameter set largely in any perturbation that we expose these cells to, the primary condition for any of these cells to have plasticity becomes the ability to access multiple phenotypes, right? Which translates to multi-stability in our system. So what we essentially do is, after simulating our networks, we look at the fraction of parameter spaces, fraction of parameter sets, or the fraction of parameter space that has more than one steady state or multi-stability. And we use that fraction as a measure of the plasticity allowed by the network, right? So obviously for each of these EMP networks, since they are allowing a switch from epithelial to mesenchymal phenotype at some or the other parameter set, every one of these networks can have multi-stable parameter sets, right? So the question that we asked is this plasticity unique to EMP networks, or could we change the network and we still get similar plasticity, right? So we want to generate some null models to answer this question. And in these null models, as you can see, what we're doing is we are keeping the degrees of each of these nodes constant. So the number of inputs, the number of outputs, everything is constant. The only thing that changes is the signs of the edges, right? So what we do is at any given iteration of generating these null models, we pick two edges in the system and just swap their signs, right? So here activation and inhibition was swapped in this manner. And we do this multiple times to generate these null models, which can be looked at as a different way in which these nodes could have connected over the course of evolution, but did not choose to connect, only connected in the way that the network was constructed. So we generated about 1,000 of these null models and calculated their corresponding plasticity scores. And this plot here shows the result of that for one of these networks, where the red line shows the plasticity for the original so-called wild type network. And the distribution here is the plasticity of all of these null models, right? And as you can clearly see, the wild type network has a higher plasticity than the null models, right? And this is the case across all the different networks that we have simulated and analyzed. So there is something unique to the network topology, which allows them to be highly plastic, which allows them to have a um, higher fraction of multi-stable parameter sets, right? So, uh, so we know that EMP networks are plastic, and we know that EMP networks are more plastic than their corresponding random counterparts. Now we move on to quantifying robustness. And a general way to define robustness is that when you make a perturbation, there is a limited change in the emergent behavior. Okay, we just like to use the term emergent behavior, which can mean different things in different contexts, but please do ignore that if it doesn't really fit the current system. Anyway, so yeah. So first of all, we need to know what kind of perturbations are we interested in understanding, and the second is what kind of behaviors are we interested in seeing the effect of these perturbations in, right? So we wanted to try to mimic what happens in biology, and there can be two kinds of perturbations, broadly speaking. One is structural perturbation, and the corresponding robustness is called structural robustness, where the network topology itself is affected, right? And so we can affect the network topology, or we can sort of mimic mutations to the network topology by simulating the like deletion of an edge, or change in the nature of an edge, or just addition of an edge as well, right? And the second kind of perturbations and the second kind of robustness that we studied is called the dynamical robustness, where the network topology itself is constant, but the parameter sets have changed, right? So in our scenario, where we are actually simulating a parameter space, not a single parameter set, we consider changes in the parameter space itself. The parameter space can shrink, it can expand, it can entirely shift to a different parameter space, and so on, right? So then what is the behavior that we're interested in? And one behavior that we see across different contexts of EMP, across different contexts of metastasis, and in fact, across different contexts of development where EMP is seen, is the relative stability of these different phenotypes, right? So as I mentioned before, there are three broad classes of phenotypes: the epithelial phenotype, the mesenchymal phenotype, and the hybrid phenotypes. And the general rule of relative stability here is that the epithelial and mesenchymal phenotypes are more stable, and the hybrid phenotypes are relatively less stable. What that means is that when given a perturbation, hybrid phenotypes are more likely to switch to a different phenotype, whereas mesenchymal and epithelial phenotype are less likely. That's what is indicated by the thickness of these arrows here, right? So we basically wanted to simulate something that, or consider something that emulates this behavior. So what we ended up doing was we ended up looking at the heterogeneity of the emergent population of cells, right? So a population of cells now, again, just to take you back, each cell has the same gene regulatory network. It just differs based on whether it's starting from different initial condition or starting from a different parameter set. And at the end, at the steady state level, we have three classes of phenotypes. We can categorize the steady state into three classes of phenotypes: epithelial, mesenchymal, and hybrid. And each of them have a different fraction. And of course, in these network simulations of EMP, we do see that the epithelial and mesenchymal phenotypes are more stable than the hybrid phenotypes, as expected from the experiments, right? So we look at the phenotypic frequency distribution or population heterogeneity and see how that changes to the different perturbations that I've described in the previous slide. Now, I'll first show you the results for the dynamic robustness aspect. And the way we are measuring the robustness is using this metric called JST. So we have the different phenotypic distributions that arise from the different kinds of perturbations that we do. In dynamic case, what we can do is we can actually compare the simulation output from the OD-based method as well as the Boolean method, where one has parameters, the other has no parameters at all, right? And what we do is we calculate this measure called JST, which is essentially telling us how separated the two phenotypic distributions are. And essentially, the lower the JST value, the lower the distance or difference between the two phenotypic distributions, and therefore higher is the robustness, right? So here, JST value is very close to 0. That means that for this particular network, the emergent outcomes of RECIPE and Boolean, the ODE-based simulation and the Boolean simulation are very same. Okay, so again, we observed this across all different networks that we simulated for EMP. And again, we asked if this is something that is unique to the network topology. We used these null models, right, or randomizations. And here we are plotting the JST between RECIPE and Boolean, between the ODE simulations and the Boolean simulations, right? And the arrows here show the JST of the wild type networks. The blue distribution showing the JST obtained from different random networks. And as you can clearly see, the JST of the wild type networks is much lower than what is observed in the JST in random networks, right? So therefore, wild type networks are more robust than the random networks. So again, network topology is crucial to enabling dynamical robustness as well as plasticity. And then we looked at the structural robustness aspect, where just to refresh again, we are doing three kinds of structural perturbations: edge deletion, edge nature change, and edge addition, one edge at a time. And this is what we generally see in all of these networks. So most of these perturbations, most of these possible perturbations end up giving you very low JST value. And there are some perturbations, of course, which are sort of very specific, which tend to push the system completely out of its original phenotypic distribution. But again, those perturbations are rare, right? So if you take an average of these different JST values and plot that for the different null models that we generated before, again, we see that the JST for wild-type networks is on the lower side as compared to JST for random networks. So for wild-type networks are more structurally robust as well as more dynamically robust. So therefore, we interpret this as EMP networks being designed to maintain a robust population heterogeneity, right? So just to give a brief summary, we looked at the phenotypic distribution and its robustness to different perturbations. And we found that phenotypic distribution emergent from EMP networks is quite robust. And EMP networks are therefore designed to enrich for robustness as well as plasticity. So next we ask, what are the common principles or what are the common structural features across these different networks? And could they be the design principles governing the emergence of robustness and plasticity? So one thing that we found, which was commonly seen across all of these networks, was an abundance of positive feedback loops. Okay, and it has been shown in multiple studies before that positive feedback loops, which are essentially feedback loops with even number of inhibitions, right? So if you start from X here, one node in your network, X activates Y activates X. So therefore, X has a positive feedback loop on itself. Similarly, two inhibitions is also considered a positive feedback loop. So X inhibits Y, but Y inhibits X. So effectively, X is activating itself, right? So we can have different kinds of positive feedback loops here of different sizes and different number of inhibitory edges. But the rule is that you need to have even number of inhibitions. And these feedback loops have been shown to be necessary for multi-stability, in that if a system is to have a gene regulator network is to have multi-stability, then there needs to be at least one positive feedback loop, right? But what we did find is that there are very general positive feedback loops in EMP networks, in that there are these mesenchymal nodes, there are these epithelial nodes, broadly speaking, and some of these nodes mutually inhibit each other, forming what is known as a toggle switch, right? So there are positive feedback loops that exist in EMP networks, and as I've said, there is an abundance of positive feedback loops, right? So one positive feedback loop is necessary for multi-stability, but why do you have so many positive feedback loops? We hypothesize that that could be the reason why these systems have high plasticity. Okay. So that's what we did. So again, we went back to the null models, the random networks, and we calculated the number of positive feedback loops in these null models as well, and compared that number with the corresponding plasticity emergent from these networks. And we see a very strong positive correlation across all different networks that we have simulated in the study. Right, that's what is shown here. You see a dominantly positive correlation for all different networks between the number of positive feedback loops and plasticity. So that is one sort of evidence that we found supporting the fact that positive feedback loops could be a reason for giving rise to plasticity, right? And when we disrupt these positive feedback loops, of course, here instead of generating random networks, we are targeting positive feedback loops in the network, again, by either deleting an edge, changing the nature of an edge, or adding an edge. And upon doing that, we again see the same thing, that the wild-type networks, which ended up having a higher number of positive feedback loops, they tend to have higher plasticity, whereas the other networks, perturbed networks, have lower levels of plasticity, right? And this is something that was also observed experimentally. So in this particular study, using CRISPR-Cas9 within mice, the researchers eliminated this particular gene, thereby effectively eliminating the positive feedback loop between ZEB and microRNA 200. And that elimination led to decrease in the metastasis observed in the mice, right? So, so we hypothesize that perturbations disrupting these positive feedback loops would reduce plasticity and can effectively reduce metastasis as well. So that's what we found in smaller networks, but we wanted to test this in larger networks as well to see if the same principle holds. Now, what we need to note here is that as the networks get larger, the number of positive feedback loops increase combinatorially. So this is a relatively smaller network with 22 nodes and 82 edges, and this particular network has over 3,000 positive feedback loops, right? And we performed the same perturbation experiment where we started changing the edges and seeing the difference in positive feedback loops and difference in plasticity. And what we see here is that the relationship is not as clean as it was in the small networks, right? So initially, for a reduction of up to 1,000 positive feedback loops, there is hardly any change in the plasticity score. But then after that, we see a sudden decrease, right? And that too, in some cases, we do see a decrease, some cases we don't see a decrease. Okay. So this tells us two things. One is that having a lot of positive feedback loops is good for plasticity, but that's not the only thing that's going to help it, right? And second thing is that we can have, so even reducing a lot of positive feedback loops might not always be enough to reduce phenotypic plasticity. We need to have, we need to make, there are additional features in the network that needs to be targeted as well. So we then looked at negative feedback loops. And the reason for looking at negative feedback loops, again, negative feedback loops are feedback loops with odd numbers of inhibition. And the reason for looking at them is because negative feedback loops are known to push the system towards monostability. They also have a lot of other features, but predominant features for smaller negative feedback loops is to push the system towards monostability. So then we thought if any of these perturbations, in addition to decreasing positive feedback loops, are also increasing negative feedback loops somehow, then the system would be pushed towards monostability. There are lesser fraction of multi-stable parameter sets and therefore lesser plasticity, right? And that's what we did observe in all of these perturbations. So remember, these are single-edge perturbations, and the difference between these two perturbations where we have equal number of positive feedback loops, but different values of plasticity, is that where there is a decrease in plasticity, we see that there is a larger number of negative feedback loops as shown in the color bar here, right? So then we adjusted our metric. Instead of just looking at the number of positive feedback loops, we looked at the fraction of positive feedback loops, which is the number of positive feedback loops divided by the total number of feedback loops in the network. And lo and behold, we see that the correlation between our metric and the plasticity score increases. So therefore, this FPC could act as one design principle for understanding plasticity. How much time do I have left?
About 10 minutes, if that's okay.
Yeah, perfect. Thank you. Great. So the summary for plasticity is that there are two network features that are important for enabling plasticity: high number of positive feedback loops and a lower number of negative feedback loops. So therefore, conversely, if we make perturbations that will reduce the number of positive feedback loops or increase the number of negative feedback loops, we push the system towards a lower plasticity zone, right? So then we then moved on to ask what is the features underlying robustness, right? And this is where, honestly, I was surprised the most, because as I said, I had been viewing these two features of plasticity and robustness as opposite behaviors, right? One is about maintaining a phenotype, the other is about changing a phenotype. So, but yeah, so again, just to give you a flashback of the structure of EMP network, we see that there are these toggle switches that are, there are a lot of toggle switches in EMP networks forming between mesenchymal nodes and epithelial nodes, right? And these, essentially these positive feedback loops are connecting epithelial and mesenchymal nodes, okay? So, to my surprise, we find that the positive feedback loops do have a say in how robust a particular network is also, but not as clearly as it was in plasticity, right? So here, if we just looked at the number of positive feedback loops in across multiple random networks and the corresponding wild type networks as well, and the corresponding dynamical JST, remember that higher the JST, lower the robustness. So therefore, we are looking at decreasing axis of robustness from bottom to top. And the biological networks, as we have shown earlier, are at the lower side of the JST spectrum, so therefore the higher side of the robustness spectrum. And there, what I would like you to notice is that there are a lot of these dots which have higher number of positive feedback loops, but also have a higher dynamical JST. Okay. Similar for structural JST, there is no immediate obvious correlation between positive feedback loops and structural JST. Okay. But when we looked at the fraction of positive feedback loops, again, we see a very strong correlation between JST and fraction of positive cycles, right? So higher the fraction of positive cycles, lower the JST and higher the structural robustness. Similarly, we found robustness of population heterogeneity. Dynamical robustness is correlated with fraction of positive feedback loops. Similarly, robustness and plasticity, in the sense that when we make a change, when we make a perturbation to the network or to the parameter space, how does the plasticity score change? That is what we measure here as a structural and dynamical robustness of plasticity. And there is a very strong correlation between the fraction of positive feedback loops and different kinds of robustness that we measured in these systems, right? So, and of course, we further modify this network to create what is known as the fraction of weighted positive cycles, which essentially weights each cycle by the length of it. So that, because the idea is that longer a positive feedback loop, lesser its effect. So we then moved on to do targeted perturbations for these networks. And as we see, the perturbations that increase FWPC weighted positive feedback loops fraction increases JST and therefore decreases robustness. Sorry, it's the other way around. So decreasing FWPC increases JST and decreases robustness, and increasing FWPC decreases JST and therefore increases robustness. I think I'm saying that right. And this is something that we observed in random networks as well. So remember, random networks have a lower fraction of positive feedback loops and therefore lower fraction of weighted positive cycles as well. But this perturbation related study holds true in that if you make perturbations that would increase FWPC in these random networks, then we'll see a decrease in JST and increase in robustness and vice versa, right? So the surprising aspect is that a fraction of positive cycles or fraction of weighted positive cycles is a metric that can support both plasticity and robustness in these networks. Now, this is the story of smaller networks. Now, when we looked at larger networks for robustness, again, we see surprising levels of robustness in larger networks. So for a small network, right, if you have one positive feedback loop, one toggle switch, then you can have two steady states, M high or E high. But for a larger network, which has 3,000 positive feedback loops, 0 negative feedback loops, 22 nodes, 82 edges, you could have had of the order of let's say 1,000,000 steady states. But when we simulate these networks using Boolean formalism, we get only 36 steady states, right? And out of these 30 steady states, there's a lot of degeneracy being introduced because of these so-called signal nodes. So effectively, we only have about 6 steady states in this network, right? Which is surprising because, again, there is a huge reduction of the state space from 4.2 into 10 to the power 6 steady states possible to only 36 unique steady states, right? So, and we also saw that just having a large number of positive feedback loops doesn't really make you more plastic, right? It's the fraction that matters. So the question that we asked is, how are these feedback loops organized in larger networks, and could they be the reason why we see such a small steady state space? And I would like to note that when you have a smaller number of steady states, each of these steady states ends up being very stable. So therefore, any dynamic perturbations that you make to the system is going to push the system back to one of these dominant steady states. Okay, so now this is a feature that we found as a higher order feature built in because of all of these positive feedback loops. And the feature is as follows. So if you look at the adjacency matrix, which tells you the interactions between individual pairs of nodes, we don't really see much, right? But when we think of sort of dynamic simulations, what the simulations are doing, what the coupled systems are doing is not only looking at the direct interactions, but also looking at the indirect interactions, right? So we wanted to do something similar at the structural level, where we asked how does one node influence the other through interactions of different path lengths, right? And that's what we did. And this adjacency matrix, which looks very sparse, turns into this thing called influence matrix, right? And this influence matrix essentially combines the effect of paths of lengths up from 1 to 10. And one very striking feature that we see is that there are these two teams of nodes based on the way the influence matrix is organized. such that each team has nodes positively influencing each other, and across the teams, nodes are negatively influencing each other, right? So this looks like a higher order toggle switch between two teams of nodes. And by putting a biological classification to these nodes, we find that one team corresponds to the mesenchymal nodes, the other team corresponds to the epithelial nodes, right? So if you remember from my earlier slides, I told you that the epithelial and mesenchymal phenotypes are more stable than the hybrid phenotypes. And at a structural level, this picture gives us a very clear reason for why that could be, because there are no teams for hybrid states. There are only teams for mesenchymal states and epithelial states, right? And another feature that I would like to highlight here is that each elements in this diagonal are corresponding to sort of self loops over a large number of path sizes, right? And the stronger the red square here, the higher the intensity of the self loop. For example, ZEB2 here, that is ZEB2 participates in a lot of positive feedback loops, and therefore that has the highest intensity. The reason why I'm telling you this is that there are nodes like Snail1 which participate in negative feedback loops, and thereby could essentially disrupt our features of plasticity and robustness. But because of all the other interactions within the network, since the overall structure has now become very cohesive, that leads to the kind of robustness that we saw in the previous slide, where you only have a smaller number of steady states, and that eventually leads to any dynamical perturbation pushing the system back to the original set of steady state and making it very robust, right? So we find, as expected, that teams provide both structural and dynamical robustness. So we define what is known as team strength, which is essentially the intensity of each of these different subrectangles that we have. And we generated null models as we did before. And random networks had a very low team strength as compared to the wild type network. So therefore, this is clearly a very unique feature to the wild type networks, more so than having more positive feedback loops or lesser number of negative feedback loops or a fraction of positive feedback loops, right? And when we make structural perturbations to this network, we see that the average structural JHD decreases with team strength. Therefore, the robustness increases with team strength. And similarly, when we make more extensive structural perturbations as well, what we do end up seeing here is that the networks with higher team strength, they respond less to these structural perturbations than the networks with lower team strength, right? So these figures show that teams do provide structural robustness. And similarly, we looked at dynamical perturbations by looking at the relative stability of each individual phenotype. And as we perturb the state of a system by flipping a fraction of nodes in their states, we see that the epithelial and mesenchymal phenotypes, which are supported by teams within the network structure, they are able to resist perturbations for a longer time as compared to the hybrid states, which are not really supported by any of the individual teams in the network, right? So this picture also explains how there could be higher susceptibility to perturbations in individual phenotypes, right? So to summarize the second robustness part, there are topological features that enable robustness in EMP, which end up being the same topological features that also enable plasticity in EMP networks. And these features at a higher order are reflected as strong teams, right? And these teams are a unique architecture of positive feedback loops. And these teams are something that we observed across multiple contexts so far, including development, including metastasis, including other diseases. And we see that whenever there are such teams, the effect shows up in being a very robust phenotypic space. And there is also plasticity, because teams allow for very specific paths of transition between the epithelial and mesenchymal phenotypes by connecting these two phenotypes. So therefore, they allow plasticity and robustness. Right. So just to conclude, we have plasticity, which is the ability to change, right? And we have the robustness, which is the ability not to change. And turns out both of these properties can be enabled by the same features. So somehow the network is allowing the phenotype to be and not to be. And metastasis, therefore, is a balance between these robustness and plasticity. Okay. I would like to leave you just with this, that we so far looked at robustness in population heterogeneity, right? But there can be robustness measured in other forms as well. One of those forms is how hybrid is your population. And as this particular study shows, the more hybrid a population, the more metastatic a population can be. So having hybridness, like robust hybridness, could be helpful for metastatic cells, right? So we looked at how hybridness changes over different networks and between random networks as well. And what we see is that higher the fraction of positive feedback loops, lower the hybridness, right? So higher fraction of positive feedback loops are going to increase plasticity, increase robustness in a phenotypic heterogeneity, but they are going to lead to lower levels of hybrid rates. So now cells which have to undergo metastasis, what would they want to do, right? They want to be plastic so that they can dynamically adapt to different perturbations or different stress conditions. But they also want to have these phenotypes which are more supportive for metastasis, such as the hybrid phenotypes, right? So there has to be some sort of tug of war happening between hybridness and plasticity in terms of the network topology that the metastatic cells undertake, right? And we believe that is why that is one of the reasons why we have such low efficiency of metastasis, is because uh, higher plasticity means lower hybridness, and therefore they don't really have the right mindset to go through metastasis. But higher hybridness, the right mindset, means lower plasticity, because so that they don't have the adaptability, right? So then the question becomes, what is the best target for therapy? Do you target plasticity? Do you target hybridness, right? So it's a problem for metastatic cells, and it's a problem for us as scientists as well. So looking ahead, we have plasticity and robustness happening together in metastatic cells. But what we need to do is find what is the relevant robustness and how best to reduce it. Yeah. So these are the studies that I talked about so far are written up in these papers, if you would like to have a look at them. And I would like to thank all of these different people that helped me conduct these particular computational experiments, and the funding agencies during my PhD. That's all I have. Sorry if I was short. If you have any questions, I'd gladly take them.